Good afternoon, and thank you for joining us today for the presentation titled The Value of SNPs in Understanding Causes of Miscarriage. All listeners today should have a Q&A box on their screen. Feel free to type in questions during the presentation, and we will answer as many of these questions as possible after the presentation. Any questions that we're not able to get to, we will answer by email. Today, our speaker is Dr. Bryn Levy, who we are excited can join us. Dr. Levy is a professor in the Department of Pathology and Cell Biology at the College of Physicians and Surgeons at Columbia University. He is also the director of the Clinical Cytogenetics Lab at New York Presbyterian Hospital and co-director of the Pathology Division at Columbia. In addition, Dr. Levy is a consulting laboratory director at Natera, and that is his conflict of interest during this talk. Now I will turn this presentation over to Dr. Levy.
Thanks, Melissa, and good afternoon to some of you. I think for some of you it may be morning, and depending on where in the world you're tuning in from, it may even be evening. So welcome to everyone. I'm excited to share with you the work that I've been doing. Actually had a long interest in looking at causes of miscarriage. And today I will go over some of the more recent studies that I have been involved in that have used the newer technologies analyzing miscarriage specimens. Whenever I give this talk, I always like to kind of get to the basics. And obviously the basics begin with the human genome. We know there are 6 billion base pairs of DNA. That is the equivalent of about 3 meters of DNA when you unwind it. And we used to think way back that there were about 80 to 100,000 genes, primarily because of the estimates of the number of proteins. But we now know that one gene can, do multiple things, and the current estimate is way, way lower, around about 15 to about 20,000 genes. We know that genes are spread across 24 different human chromosomes, and that we require one chromosome from each parent, giving us a total of the 23 pairs or the 24 different human chromosomes, 46 chromosomes per somatic cell. This is just a nice G-banded image of the chromosomes. The left you can clearly see an X and Y chromosome indicating a male, and on the right karyotype you see a female with the 2 X chromosomes. And this is exactly what we would require for normal functioning and development. So what happens when things go wrong, when you get an imbalance? And the clinical consequences of constitutional chromosome abnormalities have been understood for quite a while. We had Lejeune in 1959 demonstrating that an extra copy of the smallest human chromosome, which turned out to be chromosome 21, is responsible for those individuals with a phenotype of Down syndrome. The role that chromosomes played in the etiology of Turner syndrome, Klinefelter, and XXX was also reported that same year by Ford and Jacobs. And by end of 1969, the most common viable trisomies had been described, and the focus then turned to what the clinical consequences of structural chromosome abnormalities was. I like to show this slide, particularly when I teach the medical students, and trying to give them an understanding of the difference between genetic imbalance and chromosomal imbalance. And so we know that genetic imbalance can happen at any stage in the pathway that I've shown you here and could disrupt any effective aspect of it. The ones that we are very well versed in primarily occur in the newborn, giving us the inherited errors of metabolism. Later on, we can see things like cystic fibrosis being apparent, and even later on, Huntington's disease. And these are classically the things that we think about in terms of single gene abnormalities. However, when we talk about chromosomal imbalance, you're now talking about hundreds or thousands of genes, and this typically has a more catastrophic effect. The majority of chromosomal imbalance will have its effect in that green pathway from zygote all the way to fetus, and the majority of these are going to be lethal. A small number are going to make their way to the newborn period, and those are ones that we typically see like Down syndrome, trisomy 18-13, and then a little bit later, the ones that become a little bit more evident are those involving the sex chromosome abnormalities. Often these appear to us because of failure to reach secondary sexual characteristics or because these individuals are having issues with fertility and get a chromosome workup that reveals a sex chromosome abnormality. We know that most full autosomal trisomies and all full autosomal monosomies are so lethal that their presence in the conception is not compatible with survival. Trisomies, monosomy X, and polyploids we know are the most common abnormalities observed in spontaneous abortions. And if we actually look historically, this link between chromosome abnormalities and miscarriages has been appreciated for well over 50 years now. One of the first studies that I could find goes back to 1963. So what are the challenges when we try and analyze miscarriage specimens? One of the well-known challenges is, in fact, the risk of culture failure, which, if you look at the published studies, ranges from 10 to 40%. There is the risk of maternal cell contamination, really depending on the type of tissue you get, how it's being dissected out. And obviously, if that's not done well, you could get a lot of maternal cells when you start growing up these specimens. There's also the complexity in sometimes seeing an overgrowth of maternally derived cells. This may account for upwards of about 29 to 60% of cases, again, taking published literature that shows the level of mosaicism in the various studies that are out there. I was involved in a study that I'm going to show you in the next couple of slides, which we published in 2005, that essentially showed that the changes in the techniques for handling and processing miscarriage specimens can be a great help in decreasing culture failure rates to less than 10%. But the risk of maternal cell contamination does nonetheless remain a critical issue that impedes our ability to report a true fetal karyotype with absolute certainty. So this is a study that I was referring to. It was a study that came out of Mount Sinai School of Medicine in New York when I was still faculty over there. And essentially, if you go before the study, you'll see these types of numbers being in the literature, with about 50% of abnormalities being accounted for by trisomies, monosomy X accounting for about 20%, and there you see 15% being triploides and about 15% being tetraploid and structural abnormalities. What hasn't changed is the fact that trisomy 16 is always reported as the most common trisomy that we see in miscarriage specimens, and then trisomy 21 and 22 really close on the heels of trisomy 16. So in this study, which was from 1990 to 2002, involving over 2,000 miscarriage specimens, we diligently made an effort to segregate out, maternal versus fetal tissue. We concentrated heavily on villi. And what we found is when you change the setup and you diligently dissect out the fetal tissue, you see a difference in the proportion of the abnormalities that you analyze. And so what we saw in terms of the abnormalities, about 65% were accounted for by trisomies, 11% by monosomies, about the same amount for triploides. We saw a new category, which we refer to as multiple aneuploides, and as I'll show you, this is highly correlated with advanced maternal age, particularly those over the age of 40. And then about 5.5% of the abnormalities being accounted for by tetraploids and structural abnormalities. So if you look at the host of characters in terms of the frequency of trisomies, you'll see that trisomy is by far, through all the studies, the most prevalent. As I said, followed by trisomy 21 and 22, then 15, 18, 13, 8 and 4, 9 and 14, 4 and 20, and then you see the others. And what is apparent is almost a complete lack of trisomy 1 and 19. In 12 years, I've seen one case of trisomy 1, and in the literature, I believe there's maybe 2 or 3 cases reported, really reflecting most likely a very high incidence of, genes that are critical for development on those chromosomes. Monosomy X, as I said before that publication, you looked in the literature and you saw it accounted for about 20% of the abnormal miscarriages. And that value really came from papers published in the 80s where culture failure rates and contamination rates ranged from 11 to 30%, and the overall abnormality rates were only about 50%. So based on a review of over 7,000 successfully analyzed cases from the more recent studies, including the Mount Sinai study, the incidence of monosomy X appears to be really closer to 11%, which is almost half of what was, quoted before that. And that rate appears to be comparable or even less than the triploidy rate, which accounts for about 10 to 15% of the abnormal miscarriage specimens. If you look, and this is a table that you will find from the Genetics in Medicine study that came out of, when I was a faculty at Mount Sinai, you clearly see the influence of maternal age. This is very well understood and very well recognized. We know that you can see when you are less than 25 years of age, there is a greater than 60% likelihood that you will have a normal carrier type, whereas when you are over the age of 40, the likelihood that it's going to be abnormal is close to 80%. You can see, again, looking at this table, the likelihood of a multiple aneuploidy is going to be significantly higher over the age of 40, being about 12.5%, compared to under 25 to 29, where that is only about 2.1%. So in summary, from the Genetics in Medicine 2005 paper, modifications in physician communication and sample processing contributed to significant improvements in culture success rates, in the ratio of male to female cases with normal karyotypes, and in the percentage of cases with abnormal karyotypes. The overall abnormality rate in miscarriages appears to be, between 60 and 70%, which is far higher than the value quoted of, 50% from the 1980 papers. Monosomy X accounted for less than 10% of all abnormalities in that study, indicating that its contribution to pregnancy loss appears to be much lower than was currently believed. Multiple aneuploidy accounts for about 8% of the abnormal cases, and as I showed you, there is a very strong correlation with advanced maternal age, particularly when you get to over the age of 40. So, doing chromosome analysis following miscarriages, we know that POC testing can identify the cause. It can help determine the risk of recurrence, prevent more invasive and costly workup, trying to find out the reason. And this is particularly relevant in cases where there are greater than one miscarriage or recurrent pregnancy loss. And from my point of view, and this is dealing with many patients, I think this is actually one of the more important reasons, is helping patients understand the why of, why this occurred and will this happen again. I'm sure many of you have dealt with patients, realize that there's a lot of guilt. Questions like, did the Tylenol that I took, cause the miscarriage? I ran a marathon when I didn't even know I was pregnant, or, I had a couple of drinks at the New Year's Eve party, and again, I didn't know I was pregnant. Is this the reason that I lost the pregnancy? And, when we see a trisomy 21 or a trisomy 16, we can definitively arrest that type of guilt and give them really conclusive answers. It's very important for closure. When you lose a pregnancy, there is well-documented, a sense of loss and all grieving that comes with it. And so finding a definitive answer, in my opinion, is quite a crucial aspect for one of the reasons that doing POC testing is helpful for patients. So there is the recurrent pregnancy loss patients. We know that about 5% of fertile couples will experience 2 consecutive miscarriages, and about 1% of couples will experience 3 or more consecutive miscarriages. We know that about 5% of recurrent pregnancy loss couples carry a balanced translocation that predisposes them to loss. And if you can identify these couples, you may be able to successfully offer them added options, which include in vitro fertilization with pre-implantation genetic diagnosis. So again, genetic testing can give patients answers, and I like this information that comes from the fertility and sterility paper in 2010 that shows that 80% of miscarriages remain unexplained if you do not offer, some type of testing or karyotype or chromosome testing. And if you actually then offer chromosomes or look at the chromosomes, only 18% remain unexplained once you have done a fetal karyotype. And as I showed you, that may even be higher in certain circumstances. Between 60 and 70% of miscarriages can be attributed to chromosome abnormalities. How have people looked and studied POCs? Well, you can do traditional testing by karyotyping. That's been done for many, many years. There are people that will do fish panels, what are particularly helpful when you get culture failure, and you can look at a whole select number of chromosomes that are common in POCs. Then the more recent is looking at microarray analysis, which can be done either by array CGH or can be done by a SNP-based approach. One of the aspects that I do at Columbia, and I know that the Natera group does as well, is to use a SNP-based microarray. We found that this, when you leverage genotype information that you get from the SNPs, it adds a whole level of additional diagnostic capabilities, like your ability to detect uniparental disomy, triploidy, consanguinity, mosaicism. MCC stands for maternal cell contamination, and having the genotypes does give you the ability to look at the parent of origin. Microarray analysis in clinical genetics has really exploded over the last, I'd say, 6 or 7 years. Its diagnostic yield has been well documented in pediatric and adult patients that have intellectual disability and or developmental delay and or congenital abnormalities. I was involved in the prenatal study that came out of Columbia that again demonstrated its clinical utility in prenatal specimens. And later on, I'll speak about the clinical utility in stillbirth. Again, a big NICHD trial that was done here at Columbia together with the RTI group and the NICHD. These results often demonstrate genomic imbalances that are beyond the resolution of traditional cytogenetic G-band analysis, so you definitely see an increased diagnostic yield. If we look at chromosomal microarray analysis reports in POC specimens, they have been limited to small array CGH cohort studies that are focused on karyotypically normal products of conception, fetuses with multiple structural anomalies, or fetal specimens that have failed to grow in cultures. And prior to 2014, the literature does lack large-scale studies, greater than 2,000, assessing microarray analysis in products of conception. And so that brings us to the focus of the study that was published in Obstetrics and Gynecology in 2014. And this was a study of well over 2,000 patients. The POC samples were collected as part of a commercial chromosome microarray diagnostic test that is designed to detect ploidy of all 24 chromosomes. This was done by the Natera group as part of their commercial test. Parental peripheral blood specimens, usually maternal, accompanied most POC specimens. A missed abortion was diagnosed according to the standard at each referral site. And upon receipt at the laboratory, the villi was separated from maternal decidua as necessary via a standardized technique. The mean maternal age was 36.2 years, but you do see quite a diverse range from just over 18 to just under 50. The mean gestational age at the time of DNC was about 7.7 weeks. And again, we included only those that were not classified as stillbirth, in other words, those that range from 3 to 20 weeks of gestation. The Illumina Cyto SNP12 genotyping microarray platform was used for the microarray analysis. We chose a 10 megabase cutoff to represent the threshold detection limit of routine cytogenetic analysis in POCs. And the reason we did that is because a typical metaphase cell contains about 400 to 500 bands per haploid genome. And at this stage of chromosome condensation, deletions or duplications that are greater than 10 megabases are readily detected. So we know that in studies of miscarriages and in some prenatal specimens, banding resolution is quite an issue. You typically see banding resolutions at the 400 to 450 level or below. And so using a cutoff of 10 megabase was chosen to represent the threshold detection limit of routine cytogenetic analysis that we typically see in miscarriage specimens. So when we talk about cytogenetically normal in this study, we are specifically referring to cases that do not have imbalances greater than 10 megabase. So what did we see? And these are the results. 2,400 fresh specimens were processed, and right off the bat, what you can see is results obtained in greater than 99.875%, meaning that only 3 out of those 2,400 cases, there were no results obtained, and these are primarily because of inadequate DNA specimens coming. You see that we obtained results in 1,868 samples, representing 78% of cases, and maternal cell contamination does represent still 22% of the cases. So it shows that despite diligent cleaning, by some centers, other centers may not be as good at cleaning the villi or cutting out the villi or what they send. And so maternal cell contamination does remain an issue. If you can identify it a priori, as was done by the SNP array, you really are doing a service to the patient by not giving a false result of a normal 46XX in those types of cases. So you can identify those that are fetal, and if you focus on those, just under 60% of them showed an abnormal result. Normal results were seen in just over 40% of cases. You also look at the normal results and look at the male and female sex ratio, which turns out to be about 0.91, and this is significantly higher than the mean ratio of about 0.71 that has been reported in many large POC studies. Concentrating on the abnormal results, we see that the single aneuploidies account for the majority of these, just under 78%, with the majority being represented by trisomy, well over 92%, and monosomy only accounting for about 8% of those. If we look at the origins of these, we see that paternal origin accounts for the majority of the monosomies, and the overwhelming majority, almost 97% of the trisomies, are maternal in origin. Multiple aneuploidy, again, right in line with what we would expect from prior studies, representing just about 8% of cases. These were primarily double aneuploids in 77 cases, representing 90% of the multiple aneuploidies, and then just under 10% being accounted for by triple aneuploids. The origins are, for the double aneuploidies, again, the majority were maternal in origin, and less of a factor for the triple aneuploids, only about 62.5% were maternal. Triploidy, again, very in line with what we would expect, about 10.3%, with the majority of these being straight triploidies, and then about 14% being either a hypo, hyper, or a pseudo-triploid. If we look at the origins, the parental origins, 67 of them were digonic, and 31 were diandric, so about almost 70% were digonic and about 30% diandric. And that did not appear to be the case. In fact, that appeared to be reversed when we look at the hypo or hyper or the pseudo triploids. Partial aneuploidies, these primarily took the form of unbalanced translocations, deletions, marker chromosomes, isodicentric chromosomes. And if we look at the parental origins, we know there is not an age relationship with partial aneuploidies, and therefore we would not expect to see a greater preponderance of maternal versus paternal, and indeed that is exactly what we saw. There was no striking age effect with the partial aneuploidy. Aneuploidy plus a partial aneuploidy really represented a handful of cases, only 5 cases, and so it's kind of meaningless in terms of the parental origins, really not significant enough, but we just list them for completion of the full data set. Tetraploidy represents 4 cases, and I'm going to speak about the significance of those in a couple of slides. So here we see the full spectrum of the visible cytogenetic abnormalities. Again, it's no surprises, well in line with what we've seen before. Trisomy accounting for over 70% of the abnormalities, monosomy for about 6%, the multiple aneuploidies for 8%, and the structural anomalies for around about 4%. If we look again at the parental origin, which was clearly evidenced in the prior slides, we can see for the single autosomal trisomies that maternal origins are well over 96%. Monosomies, we did see a higher paternal origin, but that's primarily in monosomy X. We know that about 35 of the monosomies out of about the 295 cases are accounted for by monosomy 21. This is, in fact, probably the only other monosomy that makes it beyond the embryonic stage, and so we do see monosomy 21, and interestingly, about 86% of those are maternal in origin. If we look at the MCC rates by gestational age, it's clear that the earlier we get the miscarriage sent in, the higher the degree of maternal cell contamination, which does correspond to a decrease in the ability to get fetal results, as you can see from the top line there. And the later you get it, the lower the risk of maternal cell contamination and the greater the likelihood of getting a fetal result. The average rate of maternal cell contamination across all gestational ages was 22%. So what causes maternal cell contamination? So we know that when you're getting a fetal specimen or a POC specimen sent in, that the majority of it tends to be maternal. The maternal and the fetal look very similar. There are labs that use dissecting IVF microscopes contained within a hood to actually separate out the maternal from the fetal tissue. It's very helpful if you can identify villi. They do have quite a distinctive structure, but this does take some skill and experience. Depending on the collection method, if there's a DNC that's done in, under medical supervision versus a miscarriage at home and just a home collection, you're going to get much higher, incidences of maternal cell contamination. And again, depending on how you separate and sort your sorting technique. Depending on how long ago the demise occurred, you could get, necrotic tissue and less viable tissue, which does affect culturing. But as this study will show, that is less significant when you are taking direct tissue and doing molecular analysis. Whether you got the whole sample, in some institutions, the sample is split with pathology. In some cases, pathology land up sending formalin fixed tissue. These are all issues that can be addressed at some stage to decrease, culture failures and maternal cell contamination rates. And as I showed you in the previous slide, the time of gestation, maternal cell contamination is more likely to occur with earlier gestation because of the, significantly less fetal tissue. This slide, again, shows the age effect, and I'll draw your attention to the multiple aneuploidy again, seeing that this is the average age being well over 40 years of age and highly correlated with multiple aneuploidy. I mentioned tetraploidy. We know that tetraploidy caused by the failure of cytoplasmic cleavage at the first division in the zygote will not be detected using a SNP-based array because you cannot really distinguish the genotypes when you double up the, when you just have a doubling. So AA, AB, and BB essentially will look identical from AA, BB, and BB. So when you get tetraploidy that is caused by alternate mechanisms, then you can detect it. So these would include fertilization of a diploid ovum by 2 sperm, because then the genotypes will be distinguishable. And these mechanisms are thought to occur very infrequently. And as such, we only saw 4 cases of tetraploidy, which represents about 0.36% of the abnormal cases. But if you extrapolate that, we know that tetraploidy is typically found in 1% of about POCs, which would translate to about 24 samples in our study. And we detected 4 tetraploidy cases, which represents about almost 17% of the expected number. So this finding actually gives us important information regarding the mechanisms leading to tetraploid fetuses by indicating that potentially up to 17% of tetraploids may in fact arise by these alternative mechanisms. So what about those imbalances that are beyond the resolution of traditional cytogenetics? And as I said, we specifically referring to those that are less than 10 megabase in size. So what we did was review these for clinical significance by cross-referencing with relatively standard criteria using common databases of normal variants like the DGV, like Children's Hospital of Philadelphia database, in-house databases, etc. And imbalances were classified as clinically significant if their genomic coordinates corresponded to common microdeletion or microduplication syndromes, if their genomic coordinates corresponded to previously reported clinically significant phenotypes with reduced penetrance, or if the imbalance was large enough, and let's say greater than 5 megabase, to have a high probability of being deleterious because of its size alone. So imbalances were classified as benign if they did not correspond to any clinically significant phenotypes according to any of the resources that I listed in the previous slides. Imbalances that did not meet any of the 3 criteria and contained at least 1 OMIM annotated disease gene were classified as being variants of uncertain clinical significance, or as you'll see the term referred to as VOUS. So here are the partial aneuploidies. As I told you, they primarily took the form of unbalanced translocations, terminal deletions, inversion duplications, marker chromosomes, isodicentrics, and then a whole host of others. You can see in the different color coding. But I'll draw your attention to the sizes. So what I'm going to highlight in these green bars are imbalances that really one could argue are really at that threshold of detection limit. We chose 10 megabase, but 11 is not far from 10, and neither is 13.6 megabases. And depending on your banding resolution in traditional cytogenetics, it is conceivable that you could potentially miss that type of imbalance. What that means is if you miss it, if you look in the unbalanced translocation, then you change what you're actually reporting from an unbalanced translocation to a terminal deletion or duplication. And those have different reproductive consequences. The ones that are highlighted in pink now are below the resolution. So in these types of cases, if you were doing traditional cytogenetic analysis, you would most likely miss those and deliver the incorrect diagnosis because you'd be only picking up only one side of the imbalance. And the ones here in orange are basically indicating those cases where you would potentially give the incorrect diagnosis of the abnormality. We know that from the study, about 18% of partial aneuploidies may have been missed by standard G-band analysis because they had imbalances that were really at that 10 to 15 megabase kind of threshold. In some cases, as I said, a second cytogenetically visible aberration was evident, if you could argue that if the cryptic imbalance was not concurrently detected, the reported structural abnormality would not accurately have reflected the true fetal karyotype. And since unbalanced rearrangements carry reproductive implications, you need to look to see if any of the parents is a balanced carrier, and you need to initiate, testing that will determine whether or not they do carry the translocation in a balanced form. We know that upwards of 5% of couples with recurrent miscarriages carry a balanced rearrangement, and so it's imperative that such cases are discovered at the time of products of conception testing so that the appropriate parental follow-up studies can be ordered and initiated. So what about the microdeletions and microduplications in this study? And so we're going to focus primarily on those normal results, remember, below the resolution of standard cytogenetic testing. And here we saw that 33 cases, which represents 4.3%, had a microdeletion or microduplication. We looked to see that 12 of them would meet the criteria for being clinically significant. And if we look at the origins of those, about 75% of them were microdeletions and 25% were microduplications. Variants of unknown significance were found in 21 cases, representing 63%. And the majority here is actually a reverse of the clinically significant ones, being that the majority were microduplications in 80% and about microdeletions in just under 20%. The parental origins, again, these numbers are very, very small. What did seem to be showing some type of trend was the microduplications in the variants of unknown significance, with 14 out of 17 being maternal in origin. But again, these numbers are relatively low. Uniparental disomy accounted for just under 1% of cases, with 3 cases being whole genome UPD, and interestingly, these were complete molar pregnancies. 2 of them were androgenetic, and 1 was gynogenetic. Single chromosome UPD was seen in 4 cases, and here you see maternal uniparental disomy of 16, 17, and 22. And this is almost certainly a likely a result of trisomy of those chromosomes with subsequent rescue, which has been well documented in other studies. Well, what about the abnormal results? It's less interesting because almost certainly the aneuploidy accounts for the cause of the miscarriage. But if we look at the breakdown, you're going to see aneuploidy with uniparental disomy, partial aneuploidy and uniparental disomy, and aneuploidy and a microdeletion. Here we use the term clinically significant, meaning at least from the criteria, but that does not mean that they are the cause of the miscarriage. And most likely, these clinically significant microdeletions or microduplications are, in fact, incidental findings with respect to the etiology of the miscarriage. Variants of unknown significance, in that case, accounted for 16 cases or 80%. Again, the majority of those were microduplications, and the minority were microdeletions. And again, parents of origin, it's hard to say anything about those because the numbers are very small, but we did see a similar trend in the VOUS microduplications, with about 2/3 of those being maternal in origin. So here are the clinically significant copy number changes, and the top table shows you those alone, and the one at the bottom shows those that had an additional aneuploidy. So almost certainly in the bottom ones, aneuploidy is the reason for the miscarriage. And the clinically significant copy number change is almost likely incidental in those cases. But, whoops, let me go back there. What you do see that really attests to that theory is the fact that in those that did not have aneuploidy, you saw significantly larger copy number imbalances. So it is very feasible that the large size of imbalances was a potential contributor to the miscarriage, whereas when they were found together with the aneuploidy, they were far smaller and more likely to be an incidental finding. Here are the variants of unknown significance. Again, you see those being far smaller, really around about the 2 megabase range, and it's really hard in this type of study to assign, causation to those, and I really think that we are going to require significantly more studies that are going to help us to discern, do we see recurrent variants of unknown significance that we can actually start assigning causality to the miscarriage. But right now, we are lacking that type of information. So a summary and conclusion from this study: the most obvious benefit of using a SNP-based microarray is the ability to obtain a meaningful and successful result. In the cohort of 2,400 specimens, results were obtained, as I showed you, in greater than 99.9% of cases with only 3 failures. If we look at typical culture failure rates, as I showed you right in the beginning, which range from 10 to 40% in the literature, this would have translated to a failure for from 240 to about 960 patients had they had routine testing done. More importantly, the ability to segregate out specimens with maternal cell contamination identified, in 22% of the specimens, allowed us to report a meaningful and accurate result for the remainder. Elimination of maternal cell contamination cases also prevented 539 women from receiving a misleading normal 46XX result. So this study is in fact the first microarray study reporting consecutive analysis of a large number of POC cases, which was greater than 2,000, without pre-selection. It's also the first microarray study that utilized SNPs to analyze such a large cohort. To date, the POC literature has lacked information regarding the expected incidence of genomic imbalance below the routine cytogenetic resolution, which we are classifying as less than 10 megabases. The frequency of pathogenic copy number changes in cytogenetically normal cases from the recent NICHD prenatal microarray study that came out of Columbia was 1.6%. And, that is very similar to what we found in this study, which is 1.7% of patients with normal karyotype. What we find is that this finding offers us insight into the frequency with which these types of imbalances occur in the population and underscores the need for utilizing microarray technology for cytogenomic investigation of both prenatal and POC specimens. I'm going to turn my attention now to the stillbirth study that I conducted here at Columbia. This was done as part of a larger NICHD stillbirth study, and this was published in December of 2012. We defined stillbirth as fetal death at or after 20 weeks of gestation. We know that stillbirth occurs in every 160 births in the United States. The major challenges with chromosome analysis of stillbirth are very similar to those of POCs. It's the inability to establish a viable culture, which is often linked to the state that the tissue was received. Again, if the stillbirth happened quite a while before it could get to the lab, you may be dealing with fresh, with the inability to culture necrotic tissue, whereas if it was sent to the lab as a fresh specimen, you'd have a higher likelihood of getting a successful culture. And as I said, that is related to the delay from actual fetal death to the time the tissue is received in the laboratory. From previous studies, you've seen karyotype abnormalities detected in 6 to 13% of stillbirths that have a successful karyotype. And very similar to the study that I just described, using SNP arrays in POCs, the same rationale about using the 10 megabase cutoff was chosen. The methods here, specimens were collected through the Stillbirth Collaborative Research Network, where I refer to as SCREEN. There were 59 participating hospitals. We landed up with 959 women with stillbirth who were eligible. The total stillbirth babies were in fact 972 because there was the inclusion of twins. The total study number was 532, which was represented 524 pregnancies. The 290 women who did not enroll did not differ significantly from those who were enrolled with respect to age, race or ethnic group, insurance or method of payment, or gestational age at the time of delivery. The study period was from March 2006 to 2008. It was a population-based study of stillbirths in a racially and ethnically diverse cohort in 5 geographical catchment areas. Induced abortions or of a live fetus were excluded. Full participation included a maternal interview, chart abstraction, standardized post-mortem examination, placental pathological examination, karyotype analysis, collection and testing of maternal and fetal biospecimens, and women could decline any one of these components. A separate consent was obtained for future genetic testing. Biospecimens included cord blood, placental tissue, and fetal liver and muscle tissue, and karyotypes were analyzed in a university-affiliated cytogenetics laboratories. The microarray analysis was done using the Affymetrix SNP 6.0 array, which has 1.8 million probes. The array data were utilized or analyzed using the CHAS, or the Chromosome Analysis Suite software, and using Human Genome Build 18. Copy number variants of 500 kb or larger were included in the analysis. The characterization of the copy number changes was, again, very similar to the previous study, based on ACMG guidelines and interpretation reporting criteria. CNVs were classified as benign if their full length was listed in any of the 3 commonly described databases, and the remaining CNVs were classified as pathogenic, probably benign, or of unknown significance. Pathogenic CNVs had evidence of pathogenicity according to published literature, OMIM disease, or were included in the ICA, or what we used to refer to ICA pathogenic database. CNVs classified as probably benign. The variant contained no genes at all, or evidence in the literature suggested that the variant was benign, and all other CNVs were classified as unknown significance. So here are the results. 532 karyotypes done, and straight away you can see a failure rate of about just under 30%. Normals were about 64.7%, and abnormals were about 5.8%. The same number of microarrays were done on the same patients, and here you see straight away the failure rate being about 12.6%, and that is statistically significant. Normal results were received in just over 74%, and abnormals in 8.3%. And again, the abnormality rate is statistically significant, with a 41.9% increase in the yield of abnormal results. These primarily included aneuploidy as well as pathogenic copy number changes. Variants of unknown significance was seen in 25 cases. We did then look at the microarray results of those that had karyotypes, and what you see in those that failed the karyotypes, we were able to get results in quite a significant number of patients. So that means 135 out of 157 patients that failed to receive a karyotype result actually got a result from the microarray. Those that were normal, we did see some microarray failures in 43 cases, representing 12.5%. The majority were normal, as would be expected. Aneuploidy was not detected in any of those. Remember, these are normal karyotypes. But the key finding is the pathologic copy number changes, which represented 2.6% of the cases. And then 4.1% had variants of unknown significance. The abnormals, that we did see failures by microarray. There were some called normal by the microarray, and these were primarily related to cases that had mosaicism, very low-level mosaicism by karyotype. We did see additional pathogenic findings in 3.2% and variants of unknown significance as well in one case. If we look at the same type of analysis of the karyotype results from those that had microarray results, so we were able to rescue some of the cases that had microarray failures. And if we look at that, here we're talking about 45 out of the 67 patients. So only 67% could be rescued compared to the 86% that were rescued from microarray from the failed karyotypes. Of those that had normal microarray results, we saw about 30% failing by standard karyotype. Again, the majority, 70% being normal. And abnormalities, only, these are the same as shown on the left there, representing only 2 cases. Again, those by karyotype had very, very low-level mosaicism. Failures in the abnormal microarrays by karyotype were seen in 20% of cases. About 60% were abnormal, as you would expect, a high proportion, and normal in about 20%. And the variants of unknown significance failed in about 40% of karyotypes, were normal in 56%, and were abnormal in about 4% of cases. So overall, the major aneuploidies were implicated in the etiology of stillbirth, and these are the cast of characters that we've seen before: trisomy 18, trisomy 21, and monosomy X. Trisomy 18 being seen in about 30%, trisomy 21 in 30%, monosomy X in about 25%, and trisomy 13 in about 6%. And these proportions are consistent with those reported in larger cytogenetic studies, such as those by Korteweg et al. Pathogenic or VOUSs, we saw 10 deletions ranging in size from 584 kilobases to about 25.3 megabases, 28 duplications, again ranging in size from 500 kb to 2.8 megabases, and 1 stillbirth had a pathogenic deletion and duplication that was consistent with an unbalanced translocation that was actually missed by karyotyping. We did a subgroup analysis specifically of those that had, structural abnormalities. So 67 of the stillbirths had structural abnormalities. Karyotype analysis in those cases yielded results in only 45 out of the 67, so that's 67%, with 28.9% being abnormal. When we look at the microarray analysis on the same cohort, we were successful in getting a result in 60 out of 67 stillbirths, so that's almost 90%. 28.3% had aneuploidy, 5% had a pathogenic variant, and also 5% had a variant of unknown significance. So overall, microarray analysis detected more abnormalities in the structural anomalous group than did karyotype analysis, and this was statistically significant. It represented a 53.8% increase, and anomalous stillbirths were significantly more likely than non-anomalous stillbirths to have an abnormal result on microarray analysis and karyotype, and again, that was statistically significant. So in summary, the microarray analysis provides information of detection of genomic abnormalities. Obviously, aneuploidy is a great part of that, but we do see pathogenic variants, and that's statistically significant compared to karyotype analysis. It represented a 41.9 increase in the detection rate. Microarray analysis is more likely than karyotype analysis to provide an overall genetic diagnosis, and this is primarily because of its success with non-viable tissue, and this is especially valuable in analysis of stillbirth with congenital abnormalities or in cases in which karyotype results cannot be obtained. 90 more cases, representing 24%, were able to be analyzed than karyotype analysis alone. And this is a very similar theme, tying in, the previous POC study as well as the stillbirth study. The obvious advantages are very clearly seen in both studies. So that is the end of the presentation. I do want to acknowledge my fellow colleagues at Columbia University, Ron Wapner, Delia Nachum, and Vilma Agarwal, the Affymetrix group, and the NICHD group, in particular, Uma Reddy, the Natera group were outstanding in doing the SNP array on the POCs, and the RTI group who processed the stillbirth specimen. And with that, I am going to hand back over to Melissa.
Thank you so much for joining us today, Dr. Levy. We really appreciate the time that you spent. We had one question about reviewing the SNP process. Do you think that you could briefly explain how SNPs work in terms of looking at miscarriages?
That's a tough question to answer in the 1.5 minutes that I have, but it's very similar to what we do in our pediatric and prenatal studies. From the SNP arrays, you're going to look at two aspects. You're going to look at the log2 ratios, with increased log2 ratios giving you an indication of copy number, and decreased log2 ratios showing, losses, and increase showing gains. But the additional aspect that we get from the SNPs by looking at the genotypes, in very simply, if you think of two chromosomes and biallelic SNPs, you only have three possibilities, and that's AA, AB, and BB. When you have three chromosomes, the number of genotypes increases, and that would represent various possibilities, which are AAA, AAB, ABB, and BBB. So you get four possibilities when there are three chromosomes. So the concept is by simply looking at the SNP genotypes and the number of genotypes that you get an indication of the chromosomal regions, or the copy number of those regions, such that when you see four of them, it represents three copies, and actually when you see two of them, it represents one copy. When you have one chromosome, you can either be A or B, so there are only two possible opportunities. And that's the simplified 1-minute version of how you can look at the SNPs to get both copy number as well as looking at the log2 ratios for getting copy number. I won't have time to go into how you use the SNPs to look at UPD and mosaicism and all the other things that are listed.
Terrific. Thanks so much. So I think that we are out of time for questions. If you have typed in other questions, I will be happy to reply to those by email. If you have additional questions, you're welcome to reach out either to Dr. Levy for questions specific to his presentation, or to us at Natera for any other types of questions. Our contact information is here on the final slide, if that could be pulled up. So we do, again, appreciate Dr. Levy for joining us, and also appreciate all of you for taking the time out of your days to listen to this webinar. Thank you.